Hello everyone, today we're going to be going over the light reactions of photosynthesis, alright? So this is the first part of photosynthesis, and this is what creates NADPH and ATP for the Calvin cycle later on. So this is a very important preparatory stage for the Calvin cycle later on. And I just drew this little diagram here of the thylakoid space here, the thylakoid membrane on the outside, and then the stroma, of course, on the outside here. And I also include a little picture here, a little detailed picture, if you just want some labeling. Okay, so let's start and let's see what happens first. So the first thing that happens is light will hit the plant. Sunlight will reach the plant and release photons of light. And those photons of light will hit photosystems 2 and photosystems 1, but we're going to start with photosystems 2 and hit its chlorophyll pigments, and through something called resonance, where they kind of shake, you know, these chlorophyll pigments, these little green balls, they kind of shake and pass on their energy from one to another. So it's kind of like they're having a little dance party. And then it gets to these two big pigments right here, and we can call this P680. Okay? Now once the energy reaches P680, the dance party reaches P680, what's going to happen is that here I'm representing the two electrons. So what happens is once it reaches P680, it's going to release two electrons to the primary electron acceptor, and in photosystems 2, the primary electron acceptor is pheophytin. Okay? And once it reaches pheophytin, it's going to then release those two electrons onto plastoquinin, which is the first mobile carrier. And then once it reaches plastoquinin, it's going to go on to the cytochrome complex. And it's just going to pass through and go on to plastocyan, which is our second mobile carrier. And you may ask, what is the purpose of the cytochrome complex if the electrons are just passing through it? Well, the cytochrome complex is actually a proton pump. That means it's going to pump protons against their concentration gradient. And in doing so, it's going to use the free energy, right, as the electrons pass through it. It's going to use that free energy of the electrons passing through it to pump the protons against their concentration gradient from the stroma into the thylakoid space in the center. And that's from an area of low concentration to an area of a high concentration. And the reason it can do that is because it uses that energy, the free energy of the electrons passing through it. So this would be active diffusion because it's using energy. And we'll see why pumping hydrogen protons into the thylakoid space is important later on. So the only thing we need to know right now is that the cytochrome complex uses the free energy and acts as a proton pump to create a proton gradient. And then, so the electrons keep moving through. The electrons then go from plastocyan to photosystems 1. And once it reaches photosystems 1, there should actually be another beam of light here, but basically what happens again is it kind of acts like its sibling. So light reaches it again, hits the pigment molecules, they start resonance, start having a little dance party, and then it reaches these two molecules right here. And this one is P700. So in photosystems 2, it's P680. Photosystems 1, it's P700. And then, just like last time, it's going to pass those two electrons, its two electrons, to the primary electron acceptor. And in this case, there's actually a name for it, but we don't have to remember that. So it goes on to the primary electron acceptor. And just like before, it's going to get passed on to another mobile carrier. And in this case, it's ferredoxin, FD. And then once it reaches the last mobile carrier, which is ferredoxin, it will then go on to NADP plus reductase. And it will take those two electrons, a hydrogen atom, or proton, NADP plus, and create NADPH. So that's why these two electrons here are important, because NADP plus reductase will then use those two electrons to help create NADPH, one of the very important products that we need later on. And then the other product, of course, is ATP. So how is ATP made if electrons are going to the NADP plus reductase? Well, remember the cytochrome complex? We said it was creating a proton gradient, and this is why it's so important now. So we said, just to recap, as the free energy from the two electrons passing through the cytochrome complex got to it, it allowed hydrogen protons to move against its concentration gradient from the stroma into the thylakoid space here. And that created a massive proton gradient. And this guy right here, ATP synthase, will then use that proton gradient to facilitated diffusion to take those hydrogen protons, pass them through it, right? Facilitated diffusion. It's going to take those hydrogen protons from an area of high concentration to an area of low concentration. So this is now doesn't require any energy, but since they're still quite big, they still need facilitated diffusion. So this one is going to be facilitated diffusion from the hydrogen protons from the thylakoid space now into the stroma. And as the hydrogen protons are moving through ATP synthase, it's generating energy. And what ATP synthase is going to do with that energy? It's going to take ADP and an inorganic phosphate and create ATP. So this is known as oxidative phosphorylation, because we're taking inorganic phosphate and ADP to create ATP. So that's why that proton gradient was important, because since cytochrome complex did the preparatory stages, ATP synthase can take advantage of that proton gradient and create ATP for itself through oxidative phosphorylation. So now we have NADPH and ATP. But there's a couple of things that we should review, a couple notable things I want to show you. So here, let me show you this. Okay, so a couple notable exceptions, or a couple notable things to take away. Although we made NADPH and ATP, we required water. And you ask, why did we need water? Well, remember what happened when photons of light hit photosystems 2 and its P680 lost its 2 electrons and started moving through the linear electron flow? Well, those 2 electrons that were displaced, now P680 is like, where did my electrons go? So what we're going to do to fill in that void that we created is we're going to take water, H2O, and split it up. And those electrons from water is then going to fill in that disparity, so P680 is no longer lonely. It's going to take those 2 electrons and keep completing the chain. So that's why you need to water your plants. But if you look at photosystems 1, it's P700. You don't see any water here. We don't need to break up water for photosystems 1. And that's because, well, you remember those electrons that we took and brought all through this way? Well, those 2 electrons can just fill in that disparity. So those 2 electrons will come over here and fill in that disparity once these 2 electrons leave. So P700 is going to lose 2 electrons, go to the primary electron acceptor, and go on to ferredoxin and things like that. But unlike photosystems 2, instead of breaking up water to fill in that gap, we can just use the 2 electrons that came all this way to fill in that gap. So since photosystems 1 had all of this happening right before it, we don't need water. But for photosystems 2, there's nothing happening before it. So we're going to need water to get this going. And that's why you need water, but that's only for photosystems 2, not photosystems 1. And that's why. And another thing is there are two terms that we want to master. There's something called the linear electron flow, which I mentioned previously, and that was these two electrons passing from complex to complex to complex through mobile carriers, right? So take, for example, the photons come over here, resonance P680 gets excited, releases 2 electrons, goes to the primary electron acceptor, and the electrons go from photosystems 2 to plastoquinone, to the cytochrome complex, to plastocyan, to the photosystems 1, to ferredoxin, and then ends up at NADPH, right? So it goes from 1 to 1 to 1 to 1 to 1 to 1 to 1. And this is what's called linear electron flow. It's in a nice smooth line. But the other thing was, well, how much actually of this do we need, NADPH and ATP, for the Calvin cycle? Well, there's a notable disparity. We only need 6 NADPH, but we need 9 ATP. So we need a little bit extra ATP created to go on to the Calvin cycle. And how are we going to create this extra ATP? Well, remember what happened at photosystems 1? When the primary electron acceptor took its 2 electrons and finally passed them on to ferredoxin, usually ferredoxin will take them to the NADP plus reductase. But sometimes ferredoxin will go in a cyclic pattern and instead take those 2 electrons and go all the way back to the cytochrome complex. And what this does is it allows electrons to pass through the cytochrome complex again, and that gives the cytochrome complex a bit of more free energy, and this allows the cytochrome complex to pump in even more hydrogen protons and create an even greater proton gradient, right? So ferredoxin's a little bit devious here. It's going to allow the electrons to be used again, and he's a pretty clever guy. And since we create even more of a proton gradient, ATP synthase can work a lot harder and create even more ATP. So through this, going back all the way to the cytochrome complex, which we call cyclic electron flow, we can create a little bit extra ATP. And that's how we can create 9 ATP and 6 NADPH. And these two products, which are very important, will go on to the Calvin cycle, which is the next stage of photosynthesis.